You hit the alarm. 9 minutes later, again, another 9. Maybe 4 times? 5? 6? The average person who uses a snooze button presses it 2.4 times every morning. That is over 3 million nights of data from smartphone users across the globe, published in Scientific Reports in May 2025. More than half of all sleep sessions ending with an alarm end with snooze. 57% according to a Notre Dame study from October 2022, tracking 450 salaried workers. That is not laziness. That is a pattern, a global pattern. And neuroscience just caught up to what it actually does to your brain. Governs when you wake and how you think, the striatum, the prefrontal cortex, the ventral tegmental area lighting up in ways they should not, receptors downregulating, neurons firing in fragmented bursts, sleep inertia extending, decision-making collapsing. Some claim it wrecks you, some say it helps. The data published in Journal of Sleep Research in October 2023 by Tina Sundelin and her team at Stockholm University says both, and the truth sits in between. Aging Neuroscience published August 22, 2016, examining sleep fragmentation effects on dopamine production and reuptake in aging systems. From the Journal of Neuroscience, published May 6, 2012 by Volkow, and team examining dopamine receptor downregulation after sleep deprivation using PET imaging with raclopride binding, and from Neuron, published November 2, 2023 by Northwestern researchers, led by Mingjeng Wu, showing acute sleep loss increases dopamine release and rewires synaptic plasticity in prefrontal cortex. Now back to the information. The prefrontal cortex, anterior regions handling executive function, working memory, arithmetic speed. These areas take 2 to 4 hours to fully come online, according to data from a 2013 study in PLOS1 by researchers at Surrey Sleep Research Center testing 11 participants across 4 mornings. That gap is sleep inertia, the grogginess, the fog, the transitional state where you are awake but impaired. Sleep inertia hits all cognitive domains. Alertness drops hardest, sustained attention follows. Then cognitive throughput. Working memory recovers faster but still lags. Speed suffers more than accuracy. Reaction times on psychomotor vigilance tasks spike immediately upon waking, then decay asymptotically over hours. The time constant for subjective alertness is 0.67 hours. For cognitive performance, 1.17 hours. That means even after 1 hour awake, performance is still climbing back to baseline. This is not opinion. This is measured impairment, quantified in milliseconds and error rates. Primary reason cited, anxiety about oversleeping. They ran a lab study with 10 habitual snoozers, polysomnography monitoring every stage, comparing snooze versus no snooze mornings. The snooze condition involved 30 minutes of intermittent alarms. Result, prolonged waking and stage 1 sleep during the final 20 minutes. Stage 1 is the drowsy state, the lightest sleep, almost not sleep. Participants who snoozed showed worse vigor scores and longer reaction times immediately after final waking, compared to abrupt waking. Dopamine signaling spiked and fell in disjointed pulses. This is not how the wake system evolved to function. The circadian and homeostatic drives expect one clear transition. Snoozing creates multiple incomplete transitions. Snoozers were younger. They had later chronotypes, night owls. They slept shorter durations. They felt more morning drowsiness. No surprise there. Second part, 31 habitual snoozers spent 2 nights in a sleep lab. One morning they snoozed 30 minutes. Other morning they got up immediately. Full polysomnography. Cognitive tests right upon waking, arithmetic tasks, memory tasks, cortisol samples for stress hormone measurement. The snooze condition lost about 6 minutes of total sleep compared to no snooze. Not catastrophic. More importantly, snoozing prevented awakenings from slow wave sleep, also called stage 3 or N3, deep sleep. The first alarm in the snooze condition tended to catch people in lighter stages. By the time the final alarm rang, most participants had transitioned out of deep sleep naturally. In contrast, the no snooze condition sometimes ripped people straight out of slow wave sleep, which amplified sleep inertia. Waking from deep sleep produces the worst grogginess, the longest recovery times. The Sunderland team found that snoozing actually improved cognitive performance on some tests immediately after final waking, compared to abrupt waking from deeper stages. Arithmetic speed was better. Working memory showed small improvements. No clear negative effects on cortisol awakening response. No worsening of mood, no impact on overnight sleep architecture. The benefits disappeared by 40 minutes post-waking. By then, both groups performed equally. But in that critical first window, the snoozers, who were habitual snoozers, performed slightly better than when forced to wake abruptly. The study concluded that for habitual snoozers, 30 minutes of snoozing does not harm and may help during the immediate post-wake period. The difference likely sits in what stage of sleep the first alarm interrupts and how many times the person actually wakes versus drifts. The Japanese participants may have experienced more deep sleep disruption despite the snoozing. The Swedish participants, all habitual snoozers, may have naturally set alarms that avoided deep sleep, or their sleep architecture adapted over time. The neurotransmitter that governs wakefulness, reward, motivation, motor control. Dopamine neurons in the ventral tegmental area and substantia nigra project throughout the brain. The striatum, rich in dopamine receptors, drives both wakefulness and habit formation. Sleep and dopamine interact bidirectionally. Dopamine promotes wakefulness. Sleep deprivation alters dopamine signaling. But how? In August 2008, researchers published in the Journal of Neuroscience showing that one night of total sleep deprivation increased dopamine levels in human striatum. They used PET scans with carbon-11 raclopride, a radiotracer that competes with dopamine for D2 and D3 receptors. After sleep deprivation, raclopride binding decreased in striatum and thalamus. Less raclopride binding means more dopamine occupying those receptors. The brain flooded the system with dopamine to keep people awake despite mounting sleep pressure. Dopamine went up, but cognitive performance still collapsed. The dopamine increase could not compensate for the deficits caused by sleep loss. They scanned 20 healthy participants with raclopride after both rested sleep and one night of sleep deprivation. Then they gave methylphenidate, a drug that blocks dopamine transporters and raises dopamine further. The key finding, sleep deprivation reduced D2D3 receptor availability in ventral striatum. This was not just more dopamine competing for receptors, it was actual receptor downregulation, fewer receptors. The methylphenidate-induced dopamine increases were the same size in both rested and sleep-deprived conditions, indicating baseline dopamine was not elevated. Instead, the receptors themselves had decreased. Sleep deprivation, even acute, triggers this response in ventral striatum. The ventral striatum mediates reward, motivation, alertness. Reduced receptor availability there correlates with reduced alertness and increased sleepiness. The participants felt worse, performed worse, and their brains had physically changed the dopamine receptor landscape in response to one night without sleep. Not one night of total deprivation, but months, years of fragmented final sleep segments, repeated micro-awakenings, brief dopamine spikes each time the alarm fires, and you half-wake. Then a drop as you sink back into light sleep, then another spike, then another drop. The system never settles. Dopamine signaling fragments. Conserved dopamine pathways with humans. Sleep fragmentation for 4 days significantly increased expression of genes regulating dopamine production, secretion, and reuptake. Dopaminergic signaling surged. The system tried to compensate. But in middle-aged flies, 4 days of recovery sleep after fragmentation was not enough to bring those elevated levels back to normal. The fragmentation induced a persistent upregulation. Chronic elevated dopamine production can cause dopamine leakage into the cytosol, which leads to neuronal cell death through oxidative stress. The protective systems meant to shield dopaminergic neurons, the endoplasmic reticulum chaperone complexes, got overwhelmed. What started as neuroprotection became neurotoxicity with sustained activation. The hypothesis emerging from this research is that chronic sleep fragmentation might accelerate dopaminergic damage, especially in aging brains, where antioxidant systems are already declining. Continual sleep fragmentation could create a vicious cycle. Disrupted sleep damages dopamine neurons. Damaged neurons worsen sleep regulation. Worse sleep regulation fragments sleep further. The cycle compounds. Schizophrenia patients who have altered dopamine signaling show severe circadian rhythm and sleep disruptions. A systematic study by Wolf and colleagues in 2012, published in Schizophrenia Research, assessed 20 patients with schizophrenia against healthy, unemployed controls. All 20 patients had significant sleep and circadian rhythm disruption. 0 controls did. The patients showed heterogeneous phenotypes, advanced phase, delayed phase, non-24-hour rhythms, not entrained to light-dark cycles, fragmented sleep, irregular patterns. Dopamine elevation disrupts sleep. Elevated dopamine through genetic or pharmacological manipulation causes fragmented rest activity rhythms and reduced sleep in animal models. The relationship is reciprocal. Sleep disruption alters dopamine. Altered dopamine disrupts sleep. Striatal dopamine levels fluctuate across the natural sleep-wake cycle. Frontiers in Neuroscience published research in March 2019 using a dopamine sensor, DE-Lite 1.1, in mouse striatum. Dopamine release was highest during wake, lower during non-REM sleep, and intermediate during REM sleep. Dopamine uptake, the speed at which transporters clear dopamine from the synapse, was positively correlated with time spent in REM, or non-REM sleep, and negatively correlated with wake time. Sleep increased dopamine uptake, shortening the duration of dopamine transients, and this makes sense. During sleep, the brain needs less sustained dopamine signaling. During wake, dopamine must stay elevated to maintain alertness. The transporters fluctuate erratically. The receptors, faced with inconsistent signaling, begin to downregulate to protect against overstimulation. This is what Volkow found in sleep-deprived humans. And if snoozing creates micro-sleep-wake transitions repeatedly, the same downregulation could occur chronically in habitual snoozers. Northwestern University researchers, led by Mingjeng Wu and Yevgenia Kozorovitsky, published in Neuron in November 2023, showing that acute sleep deprivation in mice increased dopamine release and enhanced synaptic plasticity. They induced mild sleep deprivation and examined 4 brain regions: prefrontal cortex, nucleus accumbens, hypothalamus, dorsal striatum. Dopamine release increased in 3 of the 4. The prefrontal cortex was key for the antidepressant-like effect observed. Sleep-deprived mice showed hyperactivity, increased sexuality, and reduced depressive behavior. The mood boost lasted days. Examination of neurons revealed increased dendritic spines in prefrontal cortex. Synaptic plasticity was enhanced. The brain rewired itself in response to acute sleep loss. People sometimes feel euphoric after pulling an all-nighter. Dopamine floods the system. New synaptic connections form. The prefrontal cortex, normally engaged in careful deliberation, gets rewired for a more impulsive, reward-seeking state. This is adaptive short-term. Keeps you awake and functional during an emergency. Long-term, it is damage. Chronic sleep loss combined with this repeated dopamine surge and synaptic remodeling could contribute to impulsivity, poor decision-making, mood instability. Then it drops back as you drift into light sleep. Over months, this could train the prefrontal cortex into a fragmented activation pattern. Instead of one clean transition from sleep to full wake, you get a stuttering sequence. Decision-making suffers. Impulse control weakens. The habit loop, controlled by striatum and dopamine, strengthens around the snooze behavior itself. Habit formation relies on the striatum, particularly the caudate nucleus and putamen. Dopamine from the ventral tegmental area, and substantia nigra signals reward and reinforces stimulus-response associations. A cue, the alarm sound. A routine, hitting snooze. A reward, avoiding immediate discomfort and getting 9 more minutes. Each repetition strengthens the neural pathway. The behavior becomes automatic. The prefrontal cortex disengages. The striatum takes over. This is how habits form. The striatum does not register them as punishments linked to the snooze action. The habit strengthens unchallenged. Whether those effects are net harmful or net beneficial depends on context. Cycles with repeated dopamine spikes and receptor downregulation, the harm compounds over time. The Japanese data and the animal models of sleep fragmentation point to worsened sleep inertia and potential long-term dopaminergic dysfunction. Transition naturally out of any remaining deep sleep, then 30 minutes of snooze may help. If you sleep 6 hours and your alarms repeatedly interrupt deep sleep or REM, forcing incomplete awakenings, the fragmentation damages more than it helps. Affected. Executive functions, higher order thinking, planning, working memory, these recover slower than simple tasks. A study published in Applied Sciences in April 2021 by Italian researchers examining memory systems found that sleep inertia hit semantic memory, episodic memory, and procedural memory differently. Reaction times for lexical decisions, detecting whether a word is real, stayed slow for 20 minutes after waking. Accuracy was less affected. Speed is the sensitive measure. The brain can eventually get the right answer, but it takes longer, prolonged by fragmented waking. A PMC article examining sleep inertia in operational settings notes that performance impairments immediately after waking can equal or exceed impairments seen after 40 hours of total sleep deprivation. That is catastrophic. It does not prevent the initial period of impairment. Bright light exposure during waking helps, but blue enriched light shows limited benefit for sleep inertia compared to sleepiness. Light before waking, dawn simulation, has shown mixed results. Some studies find no benefit, others find modest improvements. Naps under 30 minutes typically stay in lighter stages and produce less severe sleep inertia. Longer naps risk waking from deep sleep. Sleep inertia from a 90-minute nap can last longer than the nap itself. Timing matters. Waking during the circadian low, the early morning hours, worsens sleep inertia. Prior sleep loss exacerbates it. Chronic sleep restriction makes every waking harder. For habitual snoozers, the Sunderland study offers some reassurance. 30 minutes of snoozing did not destroy performance. It cost 6 minutes of total sleep. It prevented deep sleep awakenings. It slightly improved immediate post-wake cognition. The cortisol awakening response, a marker of stress system activation, was unchanged. Mood was unchanged. But these were habitual snoozers tested in a lab for 2 nights. Not chronic sleep-deprived shift workers snoozing for months. Not someone hitting snooze 6 times every morning for years. But the pattern is consistent. Snoozers are more tired overall. They move less. Their sleep is worse. Whether snoozing causes this or is a symptom of underlying sleep debt and fatigue is unclear. Average number of snooze presses, 2.4. Average snooze duration, 10.8 minutes. Women used snooze more than men. Long sleep sessions, over 9 hours, were more likely to end with snooze than recommended 7 to 9 hour sessions, or short, under 7 hour sessions. People who went to bed earlier used snooze less. People who went to bed later used snooze more. Night owls, late chronotypes, snoozed more. This aligns with the Sunderland survey. Late chronotypes have circadian rhythms that favor later sleep and wake times. Society forces early waking. School starts at 8, work starts at 9. The late chronotype is chronically sleep deprived during the week. They hit snooze because their circadian drive for sleep is still high when the alarm rings. Morning drowsiness is not laziness, it is biology. The circadian low for a late chronotype extends into the morning hours. The homeostatic sleep pressure has not dissipated. Snoozing is an attempt to align social time with biological time, but it fails because 9-minute increments cannot shift the circadian phase. It is worse when waking from deep sleep, or REM. All three factors can align against late chronotypes forced to wake early. The snooze button becomes a coping mechanism, not a solution. Reducing reward sensitivity, motivation, alertness, sleep fragmentation in animal models increases dopamine production and reuptake acutely, but can lead to persistent dysregulation and potential neurotoxicity with chronic exposure. Dopamine dysregulation is implicated in Parkinson's disease, schizophrenia, addiction, ADHD, all conditions with prominent sleep disturbances. Disrupted sleep alters dopamine. Chronic snoozing, if it fragments the final sleep segment repeatedly over years, could contribute to this cycle. The striatum, bombarded with inconsistent dopamine signals during the wake transition, might downregulate receptors as a protective measure. Lower receptor density in striatum correlates with reduced wakefulness, increased sleepiness, impaired cognitive performance. The very problem snoozing is supposed to solve, morning drowsiness, could be worsened by chronic snoozing through receptor downregulation. Total sleep duration, sleep quality, chronotype, stress, diet, exercise, all of which affect dopamine systems. The study does not exist, but the biological plausibility is high, based on what we know about sleep fragmentation, dopamine regulation, and receptor adaptation. Disruption. The Sunderland data supports this for a controlled 30-minute snooze period. The Japanese data showing prolonged stage 1 sleep and worsened sleep inertia argues against it. The truth likely depends on individual differences in sleep need, chronotype, sleep stage timing, alarm frequency, and the presence or absence of chronic sleep debt. Dopamine systems are affected by sleep and fragmentation. Snoozing is a mass behavior with neurobiological consequences. Whether those consequences are net positive, neutral, or negative varies. For someone who sleeps enough and whose snoozing prevents deep sleep interruption, the immediate cognitive benefit may outweigh any hypothetical long-term receptor downregulation. For someone chronically sleep deprived, snoozing multiple times, fragmenting the last hour into a choppy sequence of incomplete wake attempts, the cumulative harm likely exceeds any benefit. Use caffeine or bright light immediately upon waking to counter sleep inertia. Avoid deep sleep interruptions. These strategies target the root causes of morning drowsiness rather than patching it with fragmented snooze intervals. For those people, if snoozing helps them feel slightly less wrecked upon waking, and they are habitual snoozers whose brains have adapted the behavior into their routine, the Sunderland data says it is probably fine for up to 30 minutes. Beyond that, the benefits fade and the fragmentation costs accumulate. If your total sleep is 7 to 9 hours, and your snoozing keeps you from getting ripped out of slow wave sleep, then snooze. If your total sleep is under 7 hours and your snoozing is stealing another 15 to 20 minutes through fragmentation, then stop. That sleep loss compounds. The dopamine dysregulation worsens. The sleep inertia extends. You start each day further in the hole. Cue. Alarm. Routine, hit button. Reward, relief. The striatum encodes this. Dopamine reinforces it. The prefrontal cortex disengages. You hit snooze before you are conscious. This is habit formation at its cleanest, and breaking it requires disrupting the loop. Change the cue, use a different alarm sound, or place the phone across the room. Change the routine. Make hitting snooze physically difficult. Change the reward. Reframe the immediate discomfort of waking as the first win of the day. If your dopamine receptors are downregulated from chronic sleep fragmentation, you have less motivation and willpower available to break the habit. The system is self-reinforcing. Snoozing downregulates receptors. Downregulated receptors reduce motivation to stop snoozing. The habit strengthens. It is a neurobiologically entrenched pattern affecting the very systems needed to change it. Breaking free requires external structure, environmental design, social accountability. Removing the option to snooze by using an alarm without a snooze function, making the wake transition non-negotiable. Others use smart alarms that detect sleep stage and wake you during light sleep. The efficacy of these varies. A multimodal bedroom alarm system tested in a PMC study in March 2024 showed modest benefits in reducing sleep inertia by using graduated light, sound, and temperature changes to ease the transition. The effect was small, but measurable. Anything that reduces the abruptness of the wake transition while avoiding fragmented snoozing could help. The 9 minutes do not provide real rest, but they provide a momentary reprieve from facing the day. That psychological function matters even if the neurobiological cost accumulates. Cultural norms that prioritize rest over productivity. These systemic changes would reduce the need for snoozing far more effectively than any individual behavior modification. Chronic snoozing could worsen the very drowsiness it aims to relieve through receptor downregulation. Acute snoozing and habitual snoozers with adequate total sleep and careful alarm timing may provide slight cognitive benefits immediately upon waking. Context determines outcome. The cumulative effect over years could be receptor downregulation, worsened motivation, extended sleep inertia, and entrenched habit loops that make waking harder, not easier. But if you are sleeping enough and timing it right, the Sunderland data suggests 30 minutes of snoozing will not destroy you. It costs 6 minutes of sleep, prevents deep sleep interruption, and slightly improves immediate post-wake cognition. The data is messy because human sleep is messy. The Japanese study found harm. The Swedish study found benefit. The American surveys found ubiquity. The animal models found dysregulation. The PETI scans found receptor downregulation after deprivation. The dopamine sensor studies found fluctuating signaling across sleep stages. All of it is true. The brain is not a simple on-off switch. Sleep is not binary. The interaction between sleep stages, circadian phase, homeostatic drive, dopamine signaling, receptor density, synaptic plasticity, habit formation, and individual differences creates a multidimensional landscape where snoozing can be harmful, neutral, or helpful, depending on where you sit in that space. Use environmental countermeasures to reduce sleep inertia. If you must snooze, limit it to 30 minutes, ensure your total sleep is adequate, and set your first alarm to avoid deep sleep interruption. Track how you feel over weeks. If snoozing helps, keep it. If you feel worse, stop. Let the data from your own nervous system guide you. The brainstem, basal ganglia, and thalamus light up instantly. Full activation within seconds. The prefrontal cortex shows delayed activation, increasing steadily over 15 minutes. The anterior regions, tied to executive function, take even longer. This temporal dissociation is why you can be awake but not alert, conscious but impaired. The lower brain systems come online fast, the higher systems lag. If snoozing fragments that activation sequence, forcing the prefrontal cortex to partially engage, then disengage, then re-engage multiple times, the net effect could be a longer time to full alertness. Instead of one smooth ramp from sleep to wake, you get a jagged staircase with multiple false starts. Each false start costs time and energy. The dopamine system oscillates instead of ramping. The receptors adapt to the oscillation by downregulating. Over months and years, this could train the brain into a fragmented wake pattern that makes mornings harder. The transition. The Sunderland data hints at this. Habitual snoozers who snoozed performed better than when forced to wake abruptly. Perhaps the habit itself, once established, serves a functional role in easing the transition for that individual. But individual differences in genetics, chronotype, sleep need, and learned adaptation patterns mean some people genuinely benefit from snoozing while others are harmed by it. The only way to know for yourself is to experiment. Track your sleep duration, snooze frequency, and cognitive performance upon waking. Run your own N equals 1 trial. 2 weeks snoozing, 2 weeks not. Measure how you feel and how you perform. Let the data from your own brain tell you what works. Use the map to navigate, but do not confuse it for the terrain. To solve. Which one applies to you depends on everything else about your sleep. Fix the sleep, fix the schedule, fix the chronotype mismatch. Then worry about the snooze button. It is a symptom, not the disease. The brain adapts to sleep deprivation by enhancing reward circuitry and plasticity. This keeps you awake, but impairs judgment. If snoozing produces microversions of this effect repeatedly, it could train your prefrontal cortex into a chronically dysregulated state. Decision-making suffers, impulse control weakens, the reward circuits misfire, the habit loop tightens. 30 minutes of snoozing in habitual snoozers, total sleep 7 to 8 hours, first alarm set to avoid deep sleep, resulted in slightly better cognitive performance immediately upon waking and no measurable harm. 6 minutes of lost sleep, no cortisol spike, no mood impact, small gains in arithmetic, speed, and memory. The benefits faded by 40 minutes, but they were real in that first window. The question is, which describes your situation? Are you the Northwestern mouse getting sleep deprived and rewiring your prefrontal cortex through repeated dopamine surges? Or are you the Sunderland participant managing your wake transition with practiced snooze timing that prevents deep sleep interruption? The difference is everything. Instead of better, stop. Set 1 alarm. Get more sleep. Address the sleep debt. The snooze button is making it worse by fragmenting what little sleep you have and down-regulating the dopamine receptors you need to feel motivated and alert. 30 minutes of snoozing did not harm their participants. It helped slightly. You're managing your wake transition in a way that works for your nervous system. The context determines the outcome. Know your context. Measure your dose, track your results, adjust accordingly. But chronic, repeated insult over months and years may push the system into a state that no longer fully recovers. The fly data showed that even 4 days of recovery sleep after 4 days of fragmentation did not normalize dopamine gene expression in middle-aged flies. Age matters. Resilience declines. What rebounds in youth may not in middle age. The antioxidant systems are stronger. The chaperone complex is more robust. The receptor turnover faster. But even in youth, training your brain into fragmented wake patterns for years could set a trajectory that makes aging harder. Sleep quality declines with age naturally. Adding habitual fragmentation on top of that decline could accelerate dopaminergic dysfunction. Parkinson patients lose dopamine neurons and suffer severe sleep disruptions. The hypothesis that chronic sleep fragmentation contributes to dopaminergic neuron loss in susceptible individuals has support from animal models. Whether hitting snooze for 10 years increases your Parkinson risk is unknown, but it is not implausible. The mechanisms exist, the pathways are there, the chronic insult accumulates. The most conservative conclusion? Minimize unnecessary sleep fragmentation across the lifespan. That includes snoozing. If you can wake with one alarm and adequate total sleep, do that. If you need to snooze to function, limit it, optimize it, and prioritize getting more total sleep so you need it less. Do not let the snooze button become a permanent crutch masking chronic sleep debt. That debt compounds. The dopamine dysregulation worsens. The wake transition gets harder. The habit loop closes tighter. Eventually, you're hitting snooze 6 times every morning and still waking up exhausted. Give your brain the transition it evolved to need. The snooze button is a modern invention. Your brain is ancient. The mismatch has consequences. Align them. The data is clear on that.